Okay, good afternoon, everyone, and thank you so much for joining us. It's my pleasure to welcome all of you to the first virtual Meet the Researchers webinar from the UK DRI and our charity founders, Alzheimer's Society and Alzheimer's Research UK. My name is Marc Aurel Busche. I'm a psychiatrist and a neuroscientist at the UK DRI at University College London. And together with my colleagues, I'd like to share with you today our excitement about dementia research, and in particular, what our researchers are doing to detect the diseases that cause dementia early in order to intervene as early as possible. Before we get started, just a few housekeeping matters. This webinar is being recorded, but only the speakers and the slides. And most importantly, we'd like to invite you to participate in this webinar. And at any point, you can ask questions anonymously if you prefer to do so, through the Q&A function at the bottom of your Zoom screen. These will be answered by the panel at the end of all the three talks. To start off, I'd like to take a moment and provide some background. I'm sharing my slides. I hope you can see that okay. So for those of you who don't know, the UK DRI is a relatively young institute and was established only 4 years ago and is the single biggest investment in dementia research in the UK, with founding funders being the Medical Research Council, Alzheimer's Society and Alzheimer's Research UK. The DRI has a unique structure and is distributed over 7 world-class research centers that are embedded in top universities across the UK. We have now grown to over 650 researchers, clinicians, support staff and students who are all equally committed to the fight against dementia. And this structure also contributes to the success of the Institute by enabling collaboration, sharing of information, and also trust among researchers, which we feel is important and key for our mission. We all feel very privileged to be part of this. There's still huge knowledge gaps in dementia, despite the importance for our healthcare systems and societies. For example, compared to cancer, there's still fewer people working on dementia, and each year there are fewer papers published on the topic, which of course means that there's much less talent, fewer ideas, and less knowledge that could translate into novel therapies. And we are really trying to change that. And the key focus of the UK Dementia Research Institute is to make fundamentally new discoveries on multiple fronts: genes, cells, brain circuits, vasculature, synapses, the role of brain inflammation, and many more, which we feel is key to solve a complex problem such as dementia. So what is dementia? Sometimes there's confusion about the use of this term. It's often used interchangeably with Alzheimer's disease, and I'm guilty of doing this as well. To clarify, dementia is an umbrella term which is used to describe disabling cognitive impairment. Several parts of cognition are usually impaired, including memory, attention, but also speech and language, route finding, decision-making, among many others. And when these cognitive problems affect your day-to-day activities, so you can't do things you're normally doing anymore, we call it dementia. And there are different diseases that lead to dementia. And Alzheimer's disease is, of course, the most common cause, the most common disease that leads to dementia. And I'm sure all of us know someone who is affected by this disease. I'd also like to highlight that there are rarer causes of dementia, for example, Huntington's disease or frontotemporal dementia. And UK DRI researchers are also world leading in trying to understand these diseases and develop novel and effective therapies. There has been a major paradigm shift over the past few years, which is the realization that dementia is really an end stage of these diseases, and that most of these diseases start many years to decades in the brain before people develop any symptoms. And here is highlighted Alzheimer's disease as one example that is probably best understood. And I hope you can appreciate that at the stage when people have memory problems or cognitive problems, which we call mild cognitive impairment, there's already a lot of change going on in the brain. There's a toxic buildup of proteins, amyloid beta and tau, which you will hear about. There are changes in brain function, brain circuits are changed. Sleep circuitry is one example that is changed early. Brain cells and their connections are often lost in areas that are important for memory and other cognitive functions. And we see that as an opportunity that allows us to detect these diseases early and then also to prevent the onset of symptoms as early as possible. And we'll hear about some of the exciting progress for the early detection today. I'd also briefly like to highlight that, of course, we're not forgetting about the millions of people who are living with dementia right now or developing dementia over the next few years. There are many programs in the UK Dementia Research Institute that target these later stages of the disease and try to find novel therapies and a better understanding. So today we're hearing about some of the very exciting progress in the early detection of dementias in the realms of genetic risk by Professor Julie Williams from the UK DRI at Cardiff, about Alzheimer's blood tests by Dr. Antoinette O'Connor from the UK DRI at UCL, and Professor Payam Barnaghi about digital biomarkers and AI to detect dementia early. He's from the UK DRI Care Research and Technology Center. With that, I'd like to invite Professor Julie Williams to share her slides. I'll stop sharing mine.
Okay, no, I'm going to talk about genes and what they can tell us about Alzheimer's disease, but more importantly, what they can tell us about the risk of getting Alzheimer's disease. So I'm going to cover finding genes. I'm going to cover a little bit on a new perspective that I think the genetics are giving us about the causes of Alzheimer's disease. I'm going to talk about the polygenic risk score, which is about predicting Alzheimer's disease. I'm going to tell you a little bit about how this might also help us in modelling Alzheimer's disease in the future. Okay, so one of the major areas that or strategies to identify genes is called genome-wide association studies. And what we do is take a sample of blood from individuals, we extract the DNA. We've given an example here of chromosome 1, and we each have 2 copies of chromosome 1, and the little red blobs indicate a variation in that DNA, those DNA strands. And what we do is actually identify these variants in individuals who have Alzheimer's disease and compare them to individuals who do not have Alzheimer's disease. And I don't know if you can see the 2 strands there, that you've got individuals with Alzheimer's in this example have more of the red versions of that bit of DNA than the grey ones, and that's the opposite way around in the normal population. And that's when we say we have association with that genetic region and Alzheimer's disease. And in genome-wide association studies, we do this for millions of genetic variants throughout the whole genome, and we do it in thousands and tens of thousands, more likely, people that have Alzheimer's disease, can compare them to those that do not have Alzheimer's disease. So it's a very simple design, and what we have found is that, and these are some of the studies, I won't go into them individually, but there are many studies that we've published over the years, and these are recent ones that are not yet published. And if you put all of these together, we have now 75 genes that affect your risk of developing Alzheimer's disease. So if I put it together, what we see is that there are patterns developing. So these genes are not random. We have more of these genes that are in immune processes and cholesterol processes and others that are coming to the fore. So there are patterns in the data that we're seeing that are giving us big clues about what may be going on in Alzheimer's disease. And I've pulled this together in what I call a multiplex model of Alzheimer's. And you see immunity is playing a very big role, and immunity has played a big role in COVID and what we've gone through for the last 18 months. But it was somewhat of a surprise to see that immunity was playing such a strong role in the common form of Alzheimer's disease that we're all familiar with. And there are other components, and I'm not going to go into these in any detail, but to point out that there are different tissues. Microglia are little organelles that occur in the brain, and they're in charge of cleaning up the brain and other functions. And there's something different about how they function that increases your risk of developing the disease. And this is what one of the messages coming from the genetics. So if you put it together, just to summarize, genes play a significant role in Alzheimer's disease, and especially the common form of Alzheimer's disease. Environmental factors do play a role, but we are interested in nailing down the genetic architecture. You can see from the figure on the right, FAD stands for the familial, very rare forms of Alzheimer's. They account for less than 1% of the disease. But you can see the common risk factors. ApoE is a very strong effect. The original GWAS loci, and I've put a line to show the new ones that we've got, these additional 75 genes. So we're beginning to explain quite a lot of the genetic variants there, but there is still quite a lot that is contributing, but we don't yet know the individual genes. But it's these that I think all of this variance we can take account of and help us to try and predict Alzheimer's disease. And this is what we use, a polygenic risk score. And we've developed this over the years. We developed it first with about 17,000 Alzheimer's cases and 37,000 controls, produced an algorithm with containing about 87,000 different genetic variants. And the bit of the table that's of interest, it's the area under the curve. And what that tells us is that if we put all of this together, we can get it right about 75 to 78% of the time. When we look at this in different populations, and I'm just going to ask you to look at the red columns here, because these relate to definite Alzheimer's disease, people that have come to pathology, and we can get it right about 84% of the time with these definite Alzheimer's cases. And when we look at those with all the risk factors, these are the ones at the extremes, we can get it right over 90% of the time. So I'm just going to move on and talk about lastly, what this can do to help us model the disease. So what we are doing at the moment is identifying individuals who have a very high polygenic risk load and individuals with a very low polygenic risk load. We're taking a sample of blood from them, identifying what we call stem cells from those samples and transforming them, changing them into cells that we're interested in, like microglia. And then we are looking at how these microglia are different in those that have most of the genetic risk factors compared to those that have the least genetic risk factors. And we think in time this is going to tell us a lot about disease mechanisms. So that's a quick run through of what we're doing with the genetics. I've sort of summarized it here, but one thing I haven't mentioned is that by doing this, we can identify people in their 30s and 40s with high risk of developing the disease. And we can really try and understand those very early stages of disease, which we do not understand at the moment. So I'm just going to say thank you. These are my sponsors, and thank you for listening. Thank you very much.
Thank you very much, Julie. If you have any questions, please feel free to put them in the small Q&A box of your Zoom screen, and we'll come to the questions at the end of the panel presentations. So our next speaker is Dr. Antoinette O'Connor. Antoinette is a clinical neurologist at the Dementia Research Center at the National Hospital for Neurology and Neurosurgery at UCLH. And as you will hear, her main interest lies in the development and assessment of biomarkers for Alzheimer's disease and related disorders. And Antoinette is also a recipient of a very prestigious Alzheimer's Society Clinical Research Training Fellowship. So Antoinette, the floor is yours.
Hi all, can you see my slides?
Okay. All right, thank you very much.
Yes, we can.
Great.
Okay.
So thanks, Mark, for the very kind introduction. As Mark's mentioned, my main interest is in developing biomarkers so that we can detect dementia earlier. And I'm particularly focusing on developing accessible biomarkers. But just to recap, I know we've talked a lot about this already today, but just a little bit of background about the progress that we've made over the last century in the road to detecting Alzheimer's disease. So we know that Alzheimer's disease was first described back in 1906 in the case of Augustine, and that was diagnosed with a clinical history of cognitive change, and then also at post-mortem when there was evidence of brain shrinkage. So you see here in the bottom of my screen an image of a normal brain, which looks quite big and chunky, and then we see what happens in Alzheimer's disease and other dementias where we see shrinkage of the brain, and we can see here that there's a big difference in the size of these two brains. And then we made some more progress later in the 1900s when we discovered what were the proteins that were driving this process, that were causing this shrinkage of the brain. And then this showed that the proteins driving it were tau proteins and amyloid. And it was thought that these processes were very important in kicking off the process that ultimately led to the clinically important problems with memory and thinking that we realize today. But the big problem was that we still could only reliably diagnose this condition at post-mortem. There was no way of reliably detecting change during life. And then in the early 2000s, a big change was the development of the MRI scanners. The MRI scanner is a reliable way of imaging the brain, and with this we can get high resolution pictures of the brain and get a better idea of what is causing symptoms. And here we can see a picture of a brain scan of a 55-year-old person living with Alzheimer's disease. And now I'm going to show you a series of pictures as time changes. So you see here this baseline, then 18 months later, and we can see here that there's subtle change in the size. And then it's 36 months later, and then 42 months later. And this shows really the importance of brain scanning that can be used to detect dementia, Alzheimer's disease, and other dementias. And it also gave us important insights into the pathobiology of disease and how the disease evolves over time. Because what was discovered was that in moderately severe Alzheimer's disease, parts of the brain have reduced in size by 30 to 40% by the time of diagnosis. And this really led to a shift towards the importance of early diagnosis when you want to intervene when there's fewer brain cells lost, so there's the most to save, and perhaps also the best chance of saving it. But in order to intervene earlier, we need easy access tools for diagnosis. We also need to be able to have specific tools for diagnosis. So earlier I mentioned about amyloid and tau protein and how we can detect those in post-mortem examination when after somebody passes away. But ideally we'd like to detect these specific proteins during life. And in the early 2000s, there was progress in this end, in that you could, the development of specific PET tracers. So a PET scan is a dedicated brain scan where a tracer is injected and it can bind to these abnormal proteins in the brain. So we see here a picture of a PET scan, and there's a normal image here on the left showing low amyloid. In contrast, in the picture on the right is a picture with high amyloids. This tracer goes up into the brain and tags all these proteins. And there we have a very specific diagnosis for Alzheimer's disease. Around the same time, CSF biomarkers were developed, and this is a test where we do a lumbar puncture, take up some fluid, and this is sent to a lab where specific proteins can be measured again amyloid proteins, and also, as we mentioned earlier, these tau proteins can also be detected. And these have revolutionized Alzheimer's disease diagnosis. They ensure that we can reliably diagnose this condition during life. However, there are some problems with these tests in that they're expensive, they're somewhat invasive, and they're not easily accessible. You often have to be referred to a tertiary referral centre. So that brings me to what a lot of my research has been on, somewhat of the holy grail of Alzheimer's disease research, and that's identifying a blood biomarker for Alzheimer's disease, so blood tests. And really there's been an explosion of progress in the last 5 years with the development of advanced immunoassay platforms. So we see here a picture of what's called a Simoa platform. And this is an advanced digital measurement technology that is 1,000 times more sensitive to the conventional measurement techniques. And from this, there's been development of some really exciting blood tests. So, you know, we've seen headlines in the New York Times, a blood test for Alzheimer's disease, is it coming? And indeed, there's good evidence that it is. There's been developments of a blood biomarker called neurofilament light. And this is essentially a measure of the structural integrity of brain cells. And we've been able to reliably measure this in blood, and it's been shown that it tracks with structural change and it tracks with cognitive change. And not only this, but it also predicts future cognitive and structural change. So this is really a really exciting measure because you can potentially see this being used in clinical trials or in real life in clinics to be used as both an outcome measure to track progress and potentially as a prognostic biomarker. However, there's one problem with neurofilament light in that it's non-specific. So it's not only increased in Alzheimer's disease and other dementias, but it's also increased in an array of other conditions that cause any injury to brain cells. So it comes back to that question of trying to detect amyloid and tau. And actually over the last 2 to 3 years, there's also been huge progress in developing these specific biomarkers. And now we have good evidence that we can detect amyloid, and that protein is reduced in the blood of those who have amyloid protein in the brain. And over the last 2 years, there's been very exciting evidence that phospho-tau, another protein we can measure in blood, and it performs almost as well as amyloid PET scans. So we really are on the frontier of having an array of blood tests that can be used to specifically diagnose Alzheimer's disease, track disease progression, and also potentially prognosticate. And this is really exciting from a research point of view because it means that it opens up clinical trials. No longer potentially will we require expensive invasive screening measures. We can instead use blood tests, and this reduces the cost of clinical trials. Also, blood tests are more easily repeatable than lumbar punctures and scans, so it completely changes the feasibility of research. But not only that, it has huge clinical implications. Blood tests are cheap and they're easy to scale up, and thus have implications for timely diagnosis. And we can imagine that a blood test can be done at point of care. And this is important because timely diagnosis is really important. It can mean that when patients and families seek help, they can get more definitive answers. It can prevent a series of investigations that are associated with harm. It allows people to enter research and trials early. And as we've seen from talks, it's very important that we intervene at an early point when there's most save in terms of cognitive function, and perhaps also the best chance of saving it. And then also it's important from a future planning point of view, and also to ensure people have access to appropriate supports, benefits, and advice. And also timely diagnosis is critical because it allows involvement in decisions when people are most able to do so. So really, blood biomarkers facilitate timely diagnosis and also improved access to research, and also potentially making it easier to participate in research. So I'd just like to thank you all for listening and look forward to taking any questions at the end. Thank you.
Fantastic. Thank you much, Antoinette. And please feel free to continue to put in questions into the Q&A box at the bottom of the Zoom screen. So our next speaker is Professor Payam Barnaghi. Payam is the Deputy Director and the Group Leader in the Care Research and Technology Center at the UK DRI. He's also the Chair in Machine Intelligence Applied to Medicine in the Department of Brain Science at Imperial College London. And his main research goal is to develop AI and machine learning solutions for healthcare and create affordable, scalable digital systems that can be applied across a range of health conditions. Payam, the floor is yours.
Thank you, Mark, for your kind introduction. As Mark said, I'm a computer scientist, and over the past few years, I have been working on developing digital biomarkers for dementia. Dementia is a neurodegenerative condition which affects people's cognitive and functional abilities. And over time, this abilities decline. But different people, obviously different groups, they will differ how this decline happens. But generally, the decline over time is not a linear sort of process. There are times which crises happen. And each time these moments of crisis happen, obviously this leads to a lot of pressure on people who are affected by dementia, and also will affect their generally their cognitive and functional abilities over time. My colleagues and I in my lab have been working on designing sort of digital technologies which we can monitor progression of the condition over time, and also use the information we collect for two key purposes. One is our primary goal is to help people who already are being diagnosed with dementia, to help to provide better, more personalized care and support to them, and also to understand how the sort of manifestation of this condition shows itself in people's behavior, in people's different activities, sleep, and in their physiology. Collecting this continuous information and understanding this will also help my colleagues, you hear from them, in their discovery science. While they are looking at molecular, cellular, genomic level, we are looking at the manifestation which affects people's day-to-day abilities. When we started this research, we had two key questions. One was what type of technologies we can actually use to collect information which is going to be clinically useful and clinically applicable. And the second is, if you're applying AI and machine learning, how are we going to use this information to make it something that's going to be useful for clinicians to make decisions, for care providers, and how this information also can be fed back to people living with dementia and their caregivers. One of the very first starting point for this was if you're designing the technology, and the technology is going to be deployed in the home, because our focus are people are working with people who are already diagnosed with dementia, to live in their home environment, and we want to help them to stay as long as possible in their home environment, be able to predict adverse health conditions which affects their health, for example, infections, sort of neuropsychiatric symptoms. And while we are doing this, also continuously look at how the progression of the cognitive impairment affects their day-to-day life, and how the changes happens in their physiology, sleep, and other behavioral activities. To do this, we needed to create something which is safe, because what we're going to collect and what we are collecting is very personal, it's very sensitive. We are interested in collecting those physiology activities in the home. The first part, when we looked around, there are obviously lots of technologies which can help available off the shelf. You can get sensors to monitor movement, get sensors which monitor, I don't know, blood pressure, body temperature, and sending information over the internet. First was how to secure them, how to create an environment which is safe and privacy aware. The second was, often when you get this off-the-shelf devices, when you get one device, you get one app. And the main challenge for us was we couldn't create like a sort of a diverse set of different applications people look. We needed to create something which we can bring all this information in one place and analyze this information to make clinically significant sort of insights from that. And on the right-hand side, I've shown just a cartoon of what type of sensors we develop. Primarily, we look at what is off-the-shelf available. Often we opt to use technologies which are passive. They don't need like changes in people's routine and day-to-day sort of activity. at the same time, we have an eye on the future. We work with different groups, some different colleagues I have in robotics, in biosensor design, to look at what the future type of sensors we can use in designing our digital biomarkers. But in principle, the data we collect are in sort of a few categories. Are environmental, like looking at ambient light, ambient temperature, movement around the house, and looking at the physiology, like blood pressure, heart rate. During COVID, we started monitoring oxygen saturation and so on. And I've just in this table, if you look at those which I have highlighted in yellow, are information we continuously collect using sensors. Most of them are passive sensors, just continuously collect the data, or depending on when an event triggers them to collect information. And then we look at some scales, like periodically we ask our participants to complete some questionnaires. We sort of do some examination to collect some gold standards sort of measurements to measure the changes in the cognitive and functional abilities. And then we use to basically align the data we have and our predictive models to see how close we can get in predicting those. We have been running this study as part of the UK DRI since 2019, but we had a previous study which was looking at this similar type of markers. This is not very up to date, but earlier this year we looked at that. We had over like 40,000 days of data we had collected, and over 20,000 now of nights of sleep data. What that data looks like, this is like one of our participants. Obviously, I've anonymized this, but it's real data in one day. On x-axis, you see the time of the day, and the y-axis, you see different markers we are collecting. And you see like at night is most of the time you can see mostly we are collecting sleep data. When people start in the morning, they usually do their physiological measurements. For example, here you see we have oxygen saturation data, heart rate, and then they go on about their day-to-day activities. And you see in main part of the house, you see the like different movement and activity during the house. And again, in the evening, you see slowly goes towards like showing them more sleep data. Now I've just zoomed in one marker throughout this was one day, now through like several months. This is around, I think, 16, 17 months. And I've taken only ambient temperature. This is the now ambient temperature in different parts of the same participant's home. And you can see how temperature it varies at different months of the year. And here now I have taken that and I've zoomed it sort of out to the entire cohort. Here I have removed on the y-axis are each line is one home or one participant, and x-axis shows again sort of duration of our study. And just quickly looking at this, you see this two yellow lines. They are like summertime if you look here, and there are heat waves. And I picked up the ambient temperature because it was very typical to UK. Obviously, most of the homes will have good insulation or heating systems, but often we don't have air conditioning. And as soon as a heating wave happens, you see the indoor temperature increases and goes very high. Now, there are studies showing increase in ambient temperature has relation to changes in people's neuropsychiatric symptoms, and especially agitation in people with dementia. But often these studies, they are looking at a single marker, and it's not just the temperature changes, people with dementia get agitated. That must have a relation to something else, changes in their physiology, changes in their sleep. And that's what looking at the holistic view and creating this digital biomarkers adds, sort of makes it interesting and adds something which you can then generalize and scale. Now, what happens if you put then all this information together? I've again picked up like two different participants in our study. On the right-hand side, you see someone's activity, like again, x-axis is time and y-axis is different months of the year. And each color, each dot will show like one sensor being triggered, one activity. Imagine if I open the kitchen door every day around the same 7 o'clock, you will see just a steady line here. And here is on the left-hand side, you see similar markers from another participants. And just by looking at this, by just visual inspection, you see there's more randomness here, and there are a lot of nighttime disturbance. Now, just collecting this data will give our clinicians a good insight of how in cases people get affected by dementia, and how like nighttime, for example, disturbance could be symptom of different conditions or could affect people's different sort of cognitive or functional abilities. But one of the reasons, for example, people could get up at nighttime, obviously could be a sort of delirium, could be people wandering around at night, or could be, for example, if they have an infection. And one of the conditions which affect people with dementia a lot are urinary tract infections. And urinary tract infections are one of the top 5 reasons in people with dementia being admitted to hospital in terms of unplanned hospital admissions. For us, understanding of sort of creating models which can predict or analyze the risk of this urinary tract infections has 2 key values. If we cannot use machine learning or sort of the data we have to triage and understand when the risk is high, then we can do a diagnostic test, like a normal standard test of like taking a urine sample and testing it. At the same time, once we start picking up more infections, we start also looking at how these infections and inflammation generally in body affects the pathology of the disease. And then the second part will help to more discovery science. At the first instance, we are also helping people to keep them, if we detect these conditions early, we try to keep them away of hospital because like if we get a urinary tract infection diagnosed early and refer people to GPs, they can, in most cases, it can be like we can use an antibiotic or we can prescribe different interventions which will stop spreading the disease. Now, how do we do this? We have created a machine learning model which looks at some existing examples. In principle, machines learn in 2 ways. Either by example or by experience. In this case, we've given some examples to the machine of previously cases we have seen people had urinary tract infections, and the machines have learned what type of patterns occur. Clinically, if you look at like if someone has a urinary tract infection, number of times they go to bathroom increases. Often this affects their sleep, and obviously if they wake up at nighttime to go to the bathroom, again, will show at nighttime disturbance. If they have an infection, in some cases, they might have an increased body temperature. And now, if you look at all the markers we are collecting, will give us this information. Now, if you feed that at a scale to a machine and train the machines carefully, they can then start learning these patterns and looking at the correlation between these patterns and give us a risk score. On the right-hand side of just taking like one participant's a sort of risk scores over time, and the red shows when we thought the person has a urinary tract infection and then the risk was high. And then if you go to the right-hand side, you see that when they start like taking antibiotics, this risk again goes down. One thing now I haven't shown is all this information becomes useful as you can present it to clinicians and in a way which they can use and take actions. For example, here I've shown in our cohort changes in vital signs. Again, each row is one participant. For anonymization, I've just put numbers against this. And these are the standards which use GPs or in hospital, they will use to say something is out of range. Like if you look at, in large cases, most of the sort of body temperature or heart rate, you see we have large number of cases which were out of range. But if you look at dementia actually affects some of these physiological measurements. Here we have put our participants against what they seem to be their norm. And one of the things, again, when we collect this information will allow us to understand these changes in the physiology and also will create more personalized way of understanding what somebody's norm is and how they deviate from that norm, then to start tracking whether there is an underlying condition which affects their health. To be able to use that in a clinical practice, in a care setting, we have created a system which feeds all this data into a secure cloud and then analyzes it using machine learning algorithms and then creates a sort of a workflow which my colleagues can intervene. We call it Minder, is a digital platform which looks something like this, and the entire information I show here is fake. We've just this is a demo version we have, which we are all synthetic data. But we have a group of nurses which they look at this data like just on a daily basis. They just log into the system starting not all day or 7 days a week. And the system, what it does, gives them all the basic vital sign data we measure, all the environmental data, and at the same time, machine learning models, they start generating different alerts. And those alerts, then we have designed clinical pathways, which our clinicians can respond to that. And all this information gets recorded in our system. Then over time, as we are also doing interventions and helping and supporting people living with dementia, we are collecting more data, which again, in return, allows us to understand the conditions better and design more accurate models and digital biomarkers for the condition. Thank you very much.
Thank you, Payam. That's a great jumping off point for the many questions we receive. And I would now invite all the speakers to share their videos. And Payam, maybe you can stop your presentation so that we can see everyone. And for the first question, and please feel free to type in questions in the Q&A box. We're trying to get to all the questions if time allows. So let me turn to you, Julie, and ask you, there's one question about genetic testing and whether it is mostly a research tool or can be used in clinical practice, and whether blood tests may add additional value to the genetic testing.
Well, in terms of what is available at the moment, NHS testing for genetics is mainly focused on identifying mutations, and it's for those that show strong evidence of having a strong familial loading of Alzheimer's disease, usually of early onset. At the moment, there isn't anything available as such medically, but there are, the internet out there and various different users may be looking to make this available. We worked with a company called Cytox, mainly because we wanted to get them to use the best algorithm, and they now have an algorithm that they can use to help predict Alzheimer's disease. But I'm not sure what context that will be used in. I think when you combine these genetic tests with other information, like blood tests, that will increase its usefulness. I will point out that there will be different stages of disease, and what may be important at an early stage may not be so important at a later stage. And these things we need to take into account. But I think amalgamating all the information of biomarkers and what you bring to the table in terms of your genetics will be a very useful way of progressing this and trying to understand who is at risk and at what stage of disease they're at.
Thank you. Antoinette, there's a question for you, whether people can get a blood test at the GP already. So is it prime time for these blood tests already? And also if you could briefly comment on blood tests for non-Alzheimer's dementias.
So yeah, great question. So the blood tests are not quite at the level where we're using them in clinical practice. They've gone from the level of being individual research labs to being replicated across multiple different labs, showing that they're reliable. And now some tests have been approved commercially for amyloid in the United States, but they're not quite at the level that we can get them at clinical practices. But it's on the horizon. If you think of the fact that 4 or 5 years ago, we could only test these proteins in lumbar punctures, and we never really thought we'd get reliable tests from blood. Things have accelerated so quickly that I think it is around the corner, but not quite there yet. And then that's a good question also about measures for other dementias, and I see the question about frontotemporal dementia. So to go back a little, one of the blood tests I measured was neurofilament light, and that is a non-specific measure, but can be quite useful as an indicator that there is something not quite right with the brain cells. So that can be non-specifically raised in many types of dementias, not just Alzheimer's disease, but also raised in frontotemporal dementia, where it's been shown to also have some prognostic utility. But again, as I mentioned, it's non-specific. Developing specific tests for the different proteins that cause the different types of dementias, like the different proteins seen in frontotemporal dementia and Lewy body dementia, that's a really active area of research at the moment. And I think it's, you know, it's not quite at the level of the arc of Alzheimer's disease, where we have these really exciting amyloid and phospho-tau blood tests, but I'm sure that it's going to follow. And there's been great progress at the DRI, particularly led by Professor Henrik Stettenberg, who I've collaborated with in my research, that is doing great work to drive this on.
Thank you. Payam, there are a couple of questions for you around privacy and monitoring people in their home environments, how they feel being monitored 24/7. Could you comment on these issues, privacy issues, how you build trust with people?
Thanks. I was typing the answer for that. That's a very, very good question. And when we started, I was very concerned about this myself, because in practice, we are creating something which monitors people, all aspects of their life on a day-to-day basis. To start with, the study, obviously, as research has received ethical approval, and we have written a study protocol. We have also done an information governance. The information, the way we store it, is pretty much, we apply the standard sort of NHS standards for the information governance, because we store also patient data and patient identifiable information. Then all the security and encryption methods are in place. People who have access to clinically identifiable information are very, very small number of people in the study who provide direct care and support. The rest of the researchers, they only get anonymized data. From the participants' perspective, we give them an information sheet which exactly we explain what devices we use, what they are used for, how long we keep their information, for what purpose. There was an issue when we started, years ago when I started this research, was the question of capacity to give consent. At the moment, we work with people at early or mildest stage of dementia who have the capacity to give consent. But over time, we are considering looking at ethical issues of because we want also to make this more inclusive, allow different people at different stages to join. And we have been working with our ethics review teams to see what is the best way of also being more inclusive towards people who have less capacity to give their consent. I can give more lengthier answer, but I guess I tried to cover some of the essential issues related to this.
Thank you, Payam. Julie, maybe this question is for you, but others, please feel free to also step in. Sometimes in families, someone is maybe affected by dementia early on, and others in the family may live up to their 90s or even older, cognitively healthy. And the question is whether genetics may play a role, lifestyle factors. Wondering about your thoughts.
Well, for the sort of common form of Alzheimer's, as I indicated, you know, this is contributed to by thousands of genes. So you share half your genes with your closest relative, but you don't share all of them. So it is most likely that if somebody has Alzheimer's or one of the dementias in your family, it's most likely that you won't get it. It's only the families where you have the very strong mutations, these very rare mutations in APP and the presenilin genes, where you usually see a number of people in the same family. So the risk is more difficult to predict because of it's the combination of genes that tip you over the edge, in combination with other environmental factors. So it's if you do have one person in your family, it doesn't mean that you're going to get Alzheimer's disease. The only proviso is if you have really early onset disease and you have a number of people in your family, then maybe you can go for genetic testing, but this is rare.
Thank you. Antoinette, I know that you're also very interested in neuroimaging, and there's a very specific question that maybe you can start to address. And the question is whether the combination of blood tests, polygenic risk score, maybe digital biomarkers, could lead to a situation that we don't need neuroimaging, MRI imaging, CT imaging anymore. What do you think as a clinician about this?
So I think it depends on what stage in someone's journey they are and what the role they're coming to a clinician for. Currently, the NICE guidelines state that you need imaging for everyone coming to you're suspecting a dementia diagnosis. And I guess the important reason we do that often is to rule out mimics, so things that can mimic or other causes of cognitive change. Sometimes inflammatory disorders can do that, or sometimes brain tumors can do that. And even so, I think there'll always potentially be a role for imaging. Now, it may not be exactly to confirm the diagnosis of dementia, but in a way to rule out important mimics.
Thank you. Payam, another question for you. You're also using EEG and sleep monitoring, and there's specific questions if brain waves change in people living with dementia, and whether you can pick that up in your monitoring systems.
That's correct. Obviously, there will be changes into that. What we are interested in is to looking at those changes in relation to what happens in their sort of behavior in their sleep. One of the key issues around this is often you can monitor EEG, for example, for a period of time. Like wearing obviously a headset to monitor EEG signals for a long time is not possible. We have a sleep group led by my colleague Professor Dekan Dek at the University of Surrey, and their main research is looking at sleep and also using sleep bands monitoring EEG and changes to these brain signals during the sleep, and also looking at how different patterns of sleep obviously change over the course of dementia, and how also sleep interventions can help sort of reduce some of the especially neuropsychiatric symptoms and generally improve the quality of life. One of the things I forgot to mention, like we've been working with one of the manufacturers of, for sleep, we are using a sleep mat, which just goes under the bed and continuously when people sleep monitors their breathing, their heart rate, and basically whether they are sleeping in bed. And we contacted the manufacturer, and we have got something around 1 million nights of sleep data from general population. Now that gives us a very large amount of data within general public to see how the sleep patterns change. We age matched that 1 million nights of sleep to the age group we have in our study. And we have something around over 140,000 nights of sleep of the same age matched group from a general public. When we looked at the changes in our participants and the control data we had from general public, there seemed to be significant changes in our participants group. And now putting that, looking at like sleep pattern, vital sign, and breathing rate, and obviously then looking at changes in the brainwave will give us a better understanding of how they get correlated. I take this opportunity. There was a question I answered and disappeared, saying what we do, then we take human out of the loop. And I totally agree. And I think that maybe I should have highlighted more. There is no intention to automate everything. All I say, we are creating digital biomarkers to allow us to become more personalized and more predictive in the health and support we provide. And as I said, our endpoint is giving these insights to clinicians to make better decisions. or for them to become more predictive, to go and do early interventions. There is no intention to take the human touch. I totally agree. That's a very important point.
Thank you. I think we have time for one more question, Julie. There's a question about the use of polygenic risk scores for non-Alzheimer's disease dementia, and if you could comment on that.
Valentina in my group has calculated a similar score for Parkinson's disease, but Parkinson's disease is less heritable. And the number of individuals that are affected by single very influential genes is higher in Parkinson's disease. So the predictive value of the test is not as good as in Alzheimer's disease, but there is a potential for some information to be useful there. But one thing we did observe, Caleb Webber, also at Cardiff, observed that individuals with the risk factors, the polygenic risk factors for Alzheimer's disease, declined more quickly when they had Parkinson's disease, but these did not affect your risk of getting Parkinson's disease. So what we're seeing are things that are crossing different sorts of dementia, and these things can be useful in a variety of circumstances.
Thank you so much. Unfortunately, we are up to the hour. There's so many more questions, so I think next time we need 2 hours for this webinar. I'm very sorry that we can't address all the questions. There are many more good questions about lifestyle and many other things. But yeah, there's the last slide. So thank you all very much for joining, and we really hope you enjoyed the seminar. We'll be sending out a short survey around just after the webinar, so please let us know your feedback. We really like to also keep in touch and engage with you. For instance, we'll be having hopefully some in-person events early next year in London. And we'll also invite you to sign up to our monthly newsletter, which you can see here. You can also find us, of course, on the internet, on Twitter at UK DRI. We're also on LinkedIn or Instagram and YouTube. On behalf of everybody, I would really like to thank the panelists for the presentations. Thank you all again for attending. We'd like to share with you one last video before you go. Have a good night and stay safe.
What would be your hopes for where dementia research is taking us?
I have many hopes. So of course, I think the ultimate aim is to find a way to prevent dementia.
This fundamental research that we're doing into brain function and how it changes in dementias will lead to life-changing treatments.
The means to actually identify dementia earlier.
Better understanding of how the disease changes over a lifespan.
So that we can come up with different treatments and therapies.
Explore more preventative interventions that can delay the onset of the disease.
So the progression of dementia.
Stop neurodegeneration very early in its tracks.
Stop the disease even before it presents.
We could stop people from ever experiencing the symptoms of dementia. That would be amazing.
And obviously dementia is such a wide and complicated set of diseases.
There's a long way to go from what we do in the lab, looking at small pieces of brain, to being able to help people living with dementia today or prevent people who might have it in the future. But we're working hard and research does work.
I believe this is only possible by doing teamwork and having many disciplines getting together to tackle this problem.
I hope that the awareness which is increasing in society will further increase.
But looking at all the work that we do in the UK DRI and elsewhere, it really is my hope that in the not too distant future we'll be able to come up with a new set of ways of treating dementia and even preventing it.
So that we can live in a world where no one has to live with dementia.
Where it has such a huge impact on people's lives.
I think that we will get there one day.
I see all these things happening at the moment, so I'm very optimistic for this field.
And I'm excited to be a part of it.